Good morning all. You must be expecting Dohar madams, Mohan, but due to some technical issues and other commitments, I have to engage this class. Instead of 10, I am taking at 9 o'clock. Okay. So this is your biochemistry class now, and hope you can understand the change in the class timings and all those things. Anyway, so let us start with this good morning quote. I will continue with my previous class, that is, we were discussing about the water soluble vitamins, and in that list, I am now going to talk about the two vitamins today, that is vitamin B6 and vitamin B5. These B6 is major vitamin, both are important metabolically. So all these water soluble vitamins that we are now learning, they are metabolically important, that we are understanding now. So this vitamin B6, the other name for which is it is also called as pyridoxine or pyridoxine. Some may pronounce it as pyridoxine, some are pronouncing it as pyridoxine. So it is nothing but vitamin B6. We have to remember pyridoxine is vitamin B6. And the major role of this vitamin B6 is in the amino acid metabolism. So it involves the reactions of both synthetic, catabolic and interconversion of amino acids. So this coenzyme form of vitamin B6 is important in amino acid metabolism, that you have to keep it in mind. Now, it exists in three different vitameric forms. One is pyridoxine, the other is pyridoxal, and the other is, one more is pyridoxamine. So it has got three vitameric forms. One is aldehydic, the other is aminic in nature. So all of these vitamins get phosphorylated and convert into pyridoxal phosphate. So ultimately, the coenzyme form of this vitamin B6 is pyridoxal phosphate, and in this form it exists or helps in the many metabolic reactions. So this pyridoxine is the major form of vitamin B6 among these three: pyridoxine, pyridoxal and pyridoxamine. This is the major form of vitamin B6 in diet, which occurs widely in both animal and plant tissues. So as these are the alcoholic, aldehydic and amine compounds. So they all chemically are pyridine derivatives. So basic ring that is present in these vitamin B6 forms of different forms of vitamin B6, basic ring is pyridine ring, to which the functional groups that is attached makes it in the different forms. So pyridine with alcoholic group becomes pyridoxine, with amine group becomes pyridoxamine, and with aldehydic group these functional groups are different, and that makes the vitamin form different. So these are basically they are the derivatives of pyridine ring with different functional group attached to that pyridine ring. So aldehyde group of PLP forms aldimine derivatives with primary groups of the amino acid. So this aldehyde group of this pyridoxal helps in formation of the aldimine derivatives with primary groups of the amino acid. So amino acid has got amino and carboxylic groups apart from the side chain groups, so that will be involved in the reactions that are you are going to understand in amino acid metabolism in the next classes. So this active form of vitamin pyridoxine is pyridoxal phosphate. That means there is phosphorylation with the help of pyridoxal kinase in presence of utilization ATP. In the utilization of ATP, the reaction which converts this pyridoxine to pyridoxal phosphate is catalyzed by the enzyme pyridoxal phosphate in pyridoxal kinase in presence of ATP. So for this particular phosphorylation, ATP is being utilized, and the main source of this pyridoxal kinase, it is the human brain, which is found to be the richest source of this kinase enzyme. The conversion takes place like this. So this is the pyridoxine, different forms, doxyl, doxamine, all they undergo the catalysis, phosphorylation with the help of kinase, and with the help of ATP, it will be converted into its active form, that is pyridoxal phosphate, pyridoxamine phosphate, and pyridoxic acid. These are the three forms. So this is the excretory form. From this pyridoxal phosphate and pyridoxamine phosphate, ultimately it is getting excreted in the form of its oxidized form, that is pyridoxic acid. So main functional pyridoxal phosphate. So you have to remember this is a coenzyme form of vitamin B6, which is involved in metabolic reactions. Now further, let us understand how this vitamin is metabolized in the body. So absorption, transport, and excretion of this vitamin. So B6 is absorbed rapidly from the intestine by passive diffusion, and out of around 80% of ingested vitamin is absorbed. So next is phosphorylated pyridoxamine vitamin are hydrolyzed by intestinal membrane alkaline phosphatases, and dephosphorylated forms are absorbed. As I have told, in the circulation, the major form of vitamin B6 is pyridoxal phosphate. So this is the circulation. You will find this vitamin B6 in the form of pyridoxal phosphate in the circulation. This is produced from the absorbed pyridoxine as pyridoxamine. This is mainly excreted as pyridoxic acid in urine formed by aldehyde oxidase enzyme. So next excretion is in the form of pyridoxic acid after metabolism. This is being excreted in the form of pyridoxic acid in the urine. So there is limited storage of pyridoxine. Most of the body stores are associated with the enzyme glycogen phosphorylase, for which this is a major coenzyme. So out of 70% total PLP content of the body residues in muscles in association with the enzyme. So TPP is mainly involved in carbohydrate metabolism that we have seen. That is, TPP is the coenzyme form of vitamin B1. On the contrary, this pyridoxal phosphate is involved in the amino acid metabolism. So you can remember, thiamine, that is B1 coenzyme, is mainly involved in carbohydrate metabolism, whereas PLP is mainly involved in amino acid metabolism. So the function of this coenzyme function, PLP, serves as a coenzyme for broad range of amino acid metabolism, where it seems to play a central role, and hence the demand of vitamin B6 increases with the protein intake. Naturally, you can think of this. We can logically say that if the intake of protein is more, the requirement of vitamin B6 also will be more, because most of the reactions of amino acid metabolism are dependent on the coenzyme form of vitamin B6, that is pyridoxal phosphate. And hence we can say about vitamin B1 that its intake is increasing with the carbohydrate intake. As it is going on increasing, demand of vitamin B1 increases with carbohydrate rich diet, whereas demand for vitamin B6 increases with protein, more and more protein intake. So we can summarize here the biochemical role of this vitamin by understanding the following reactions. So most of the reactions which are dependent on this coenzyme form of vitamin B6, that is pyridoxal phosphate, they can be, first is transamination, the major reaction of amino acid metabolism, that is the transamination reaction. So these reactions are catalyzed by the group of the enzymes called as aminotransferases. So second group of the second class of the enzyme in the enzyme classification, if you remember, first is OTHLIL. If you remember that, the second class of the enzymes, that is transferases, and among them these amino transferases, that means the enzymes which transfer the amino group from one substrate or one enzyme to the other enzyme. These amino acid transferases, they require PLP as the coenzyme. So first group of the reaction which are dependent on PLP is the transamination reaction, and this reaction has got more and more importance that you will be understanding in detail in the amino acid metabolism. So these amino transferases more mainly clinically important. ALT and AST, they are diagnostically important enzymes that you will be learning in detail in the amino acid metabolism. Here I want to tell you that these amino transferases are dependent on PLP as a coenzyme, and this PLP is nothing but the coenzyme form of vitamin B6. So the reaction catalyzed by these transferases can be given as alanine plus alpha ketoglutarate, converting it into pyruvate and glutamic acid, and the enzyme that catalyzes is alanine transaminase. So this substrate, from this substrate, the amino group is going to be transferred to alpha ketoglutarate, converting it into pyruvate and glutamic acid. So this is a reaction which is dependent on PLP. First reaction, that is transamination. Second, now this mechanism of this reaction, always it goes like this. This PLP and amino acid, this it will accept the amino group in the first step of this amino transferases or the transamination reaction. This amino group from amino acid is taken up by the PLP, that is pyridoxal phosphate, and there is formation of an intermediate called as a Schiff base. So linking of this amino group to pyridoxal phosphate causes formation of Schiff base. This is further split into keto acid, and the pyridoxamine phosphate is separated. So conversion of this alanine takes place into keto acid. So this is the mechanism of transamination by formation of the Schiff base. So this is dependent on PLP. Second group of reactions that are dependent on PLP, they are decarboxylation reactions of amino acid metabolism. So these decarboxylation metabolism, important enzymes which are dependent on PLP, mainly they are glutamate decarboxylase and dopa decarboxylase. Now the importance of these two enzymes is that they are involved in the formation of production of GABA and catecholamine neurotransmitters in the nervous system. So that is the more important role of these decarboxylation reactions of amino acids. So glutamate will be converted into gamma aminobutyric acid, that is GABA, which is an inhibitory neurotransmitter, and that is why the deficiency manifestation of vitamin B6, you will see there is involvement of the neuropathies or neurological symptoms are seen due to vitamin B6 deficiency, as it has got role in the conversion of glutamate to GABA, that is the inhibitory neurotransmitter. So we will see further. Next is histidine will be converted into histamine by decarboxylation of histidine by the decarboxylase enzyme. So this histamine is the mediator of allergy. 5-hydroxytryptophan is converted into serotonin by decarboxylation. Cysteine is converted into taurine. Serine is converted into ethanoate. So these are all the examples of decarboxylation reactions of amino acids. So these all are dependent on PLP, that is pyridoxal phosphate. In the next following classes of the amino acid metabolism, you will come across the general reactions of the amino acids, that is transamination, decarboxylation. Next is the metabolism of the sulfur-containing amino acid is dependent on PLP. So first step, this is reaction catalyzed by cystathionine synthase in the conversion of homocysteine and serine to cystathionine dependent on PLP. Next example of the sulfur-containing amino acid metabolism is cystathionine, which be converted into homoserine and cysteine by the enzyme cystathionase. So the enzyme cystathionine synthase and cystathionase, they are requiring PLP as a coenzyme, and hence in the deficiency of vitamin B6 there is homocystinemia. There will not be utilization of or conversion of homocysteine to cystathionine is hampered, and hence there is homocystinemia, and that is why we have to use vitamin B6 therapeutically for the treatment of homocystinemia. Next important reaction which is dependent on PLP is in the synthesis of heme. If you remember, the first step in heme synthetic pathway where glycine combines with succinyl-CoA for the formation of ALA, that is aminolevulinic acid, that is catalyzed by aminolevulinic acid synthase. So this enzyme is dependent on PLP, and hence if there is lack of vitamin B6, its coenzyme form will not be there, and that is why in the deficiency of vitamin B6 we see anemia in the patients. So that is the reason, because first step in the heme synthesis is affected, there will not be formation of heme, and that will ultimately lead as a manifestation of vitamin B6 deficiency anemia. Next role, yesterday only I have told the necessity or dependence of one vitamin on the other while you understanding about the niacin, which is synthesized from tryptophan. So the important enzyme of this kynurenine pathway in the conversion of tryptophan to niacin, that is kynureninase. This enzyme requires PLP. So this is recall. You have to recall yesterday's what we have learned in the niacin formation of niacin, that PLP is required for synthesis of niacin from tryptophan. That is in the step which is catalyzed by kynureninase in that pathway, that is 3-hydroxykynurenine converting into 3-hydroxyanthranilic acid. So this kynureninase is dependent on PLP, and hence in B6 deficiency there is dermatological manifestation which is mainly because of lack of niacin. So you will see pellagra-like symptoms also in vitamin B6 deficiency. That the basic reason is there is lack of or conversion of tryptophan to niacin is hampered in vitamin B6 deficiency, and hence the deficiency manifestation involves dermatological impairments or pellagra-like symptoms that we will have to remember. Next is role in the glycogenolysis. So the phosphorylase enzyme, that is which converts this glycogen to glucose phosphate, requires PLP. So it has got role in all metabolic reactions, right from amino acid major role to these things like vitamin formation, heme synthesis, and glycogenolysis. So after understanding its metabolic role, it now becomes easy for us to understand the deficiency manifestations of pyridoxine. As such, these dietary deficiency of this vitamin is rare in human, but only it is seen in certain people, that is women who are taking oral contraceptives, the persons who are chronic alcoholics, and in some infants who are born to vitamin B6 deficient mothers. So in them you will find the deficiency manifestations of pyridoxine or vitamin B6, and because of its involvement in all the metabolic reactions that we have understood earlier, the deficiency leads to neurological, hematological and dermatological manifestations in the vitamin B6 deficiency. Now, understanding the role or underlying cause of the deficiency of vitamin B6 leading to neurological manifestation can be stated like this. The B6 deficiency leads to decreased formation of biogenic amines like serotonin, epinephrine, norepinephrine and GABA. And because of the lack of these, this leads to neurological deficiency manifestations like irritability, nervousness, mental confusion, peripheral neuropathy and depression in the patients. So apart from that, PLP is involved in the synthesis of sphingolipids. So B6 deficiency leads to demyelination of the nerves and we will find there is consequent neurological symptoms. Next, hematological manifestation, as I have told, because the first step in heme synthesis is hampered, there will be hypochromic microcytic anemia in the lack of this in the deficiency of vitamin B6 due to inhibition of heme biosynthesis. Next, dermatological manifestations mainly because of deficiency of B6 in turn leading to deficiency of niacin that manifests as pellagra-like symptoms. Apart from that, impaired antibody formation leads to carpal tunnel syndrome due to synovial swelling of carpal tunnel. And then you will see in these patients irregular indentation in the nails. The picture can be seen here. You will find here in the patients irregular indentations. This is the dermatological, or you can say one of the manifestation of vitamin B6. Apart from this, another metabolic disorder that you will find is there will be xanthuronic aciduria and homocystinuria in deficiency of vitamin B6. We have seen the roles of these two. The deficiency can be also seen due to the use of certain drugs. So following drugs have got effect on the vitamin B6 leading to its deficiency. So this isoniazid or isonicotinic acid hydrazide which is used in the tuberculosis. Yesterday also we have seen that in the one of the causes for niacin deficiency, that is use of this drug. It inhibits formation of PLP and causes vitamin B6 deficiency. Next, cycloserine acts as B6 antagonist, that is also going to affect this B6. Mild vitamin B6 deficiency may be seen in women taking oral contraceptive pills. Next, those in chronic alcoholics, ethanol is converted into acetaldehyde. This acetaldehyde inactivates PLP and hence you will find B6 deficiency neuritis in the chronic alcoholics. So this is the underlying cause of neuritis in alcoholic patients. And these drugs are found to be antivitamins, INH, deoxypyridoxine, methoxypyridoxine. They are found to be the antivitamins for vitamin B6. Now, dietary sources, all these rice polishing, germs, wheat germs, cereals, legumes, oil seeds are the richest sources of this vitamin. The requirement of this is related to protein intake and not dependent on calorie intake. On an average, adult needs 1 to 2 milligram per day during pregnancy and lactation. Requirement is up to 2.5 milligram. In overdoses, this vitamin may lead to the toxicity, and that is going to lead to sensory neuropathy The toxic effects of overdose of vitamin B6, excess use of vitamin B6 in the women of premenstrual syndrome is associated with sensory neuropathy. Some workers have suggested that vitamin B6 more than 200 may cause neurological damage. So, excess intake of vitamin B6 is going to be neurotoxic, that we have to remember. So with this we come to the end of vitamin B6, important vitamin in the amino acid metabolism in the form of pyridoxal phosphate. The reactions you have to remember here is transamination, decarboxylation. Next, its role in heme synthesis, synthesis of niacin, and apart from that, other sulfur containing amino acid and so on and so forth. Apart from this, there are the tests for detection of the pyridoxine status that can be done by assessing the enzyme, that is aminotransperases mainly. You can assess the levels of pyridoxine, indirect measure of this pyridoxine status. Next is the tryptophan load test can be performed, which involves the measurement of urinary excretion of xanthurenic acid following a low dose of tryptophan, and that will lead to tryptophan metabolic end product, that is xanthurenic acid excretion is found to be increased in B6 deficiency. So tryptophan load test is again one of the tests for detection of pyridoxine. And then you can measure the urinary homocysteine and cystathionine after a methionine load, and urinary pyridoxic acid and PLP level in blood are also going to be the important test for assessment of pyridoxine status in the blood. Next, the vitamin B5, which is also called as pantothenic acid. The chemical name of this, vitamin B5 is pantothenic acid. This is name is given because the if you break this term panto and thenic, pantos means occurrence in everywhere. So the name suggests that this vitamin is widely distributed in the nature. So that is why the name is given as pantothenic acid. You will find it is present in the nature widely. So the pantothenic acid is the derivative, or it is containing beta alanine and D-pentoic acid in amide linkage. This was first discovered that Lippmann found for the first time that it is the derivative. It is present as a part of coenzyme A in the year 1946, for which he obtained Nobel Prize in 1953. So this pantothenic acid and beta-mercaptoethanolamine are the parts of coenzyme A, and this coenzyme A is important metabolically. So pantothenic acid has got metabolic role in the form of coenzyme A. So we have to concentrate on the metabolic role of this pantothenic acid, the role of coenzyme A, that is mainly involved in activation of many biomolecules. So this is the building block of coenzyme A and of phosphopantothene, which is present in the fatty acid synthase complex. So it is related to fatty acid synthase complex. So this coenzyme a significant role in several body reactions. The biosynthetic pathways where thiol group of coenzyme A forms thioester bonds with many organic acids. So activation or the many biosynthetic pathways start with the formation of this activated organic or activated compounds, and that is by the linkage of thiol, where the thiol group of coenzyme A forms a thioester bond with the organic acid, and this results in activation of organic acid. Well known or the best two important compounds that are involved in many metabolic processes or pathways, there is the activation of successive to succinyl-CoA, acetate to acetyl-CoA. These are the two important activated acids. They are important in many biosynthetic pathways. So succinate will be converted into succinyl-CoA, and this succinyl-CoA is a precursor for heme synthesis, which you have already learned. Next, this acetate is converted into acetyl-CoA by coenzyme A, and that acetyl-CoA now is the precursor for lipid synthesis. That is, it is involved in the lipogenesis, formation of ketone bodies. So while understanding lipid metabolism, you will come to know about that. So these two are the important forms of CoA that are involved in biosynthetic pathways. So this pantothenic acid has got a complex structure containing pantoic acid, beta-alanine, and that is pantothenic acid. Now from this, coenzyme is formed by this cyclic process, converting pantothenate to 4-phosphopantothenate, 4-phospho anthothenoyl acetylene for phosphoanthothene, D-phospho-CoA, and ultimately it will be converted into coenzyme. So pantothenic acid in the form of coenzyme is important biochemically. So this is the complicated structure of coenzyme A. When it is the acyl group is added to this, it becomes acetyl-CoA or acyl-CoA. Now, the biochemical role of this coenzyme of pantothenic acid, that is acetyl-CoA, when combines with choline, it is going to be converted into acetylcholine by the enzyme acetylcholine synthase, and this acetyl-CoA will be converted into acetylcholine. CoA is released free. Pyruvate will be converted into acetyl-CoA by the pyruvate dehydrogenase. So the derivatives of CoA, acetyl, succinyl, HMG, acyl-CoA, they all are important in the metabolic pathway. So coenzyme A is an important component of fatty acid synthase complex. Acyl carrier protein also contains pantothenic acid. So these, you have to remember the role of this pantothenic acid in these formation of the CoA derivatives. So you can see how this acetyl-CoA is important. This is going to maintain the acetyl-CoA pool. We can say it is coming from fatty acid, pyruvate, and amino acids, and can be utilized for TCA cycle, fatty acid synthesis, cholesterol, and from cholesterol, steroid synthesis, ketone body formation, acetyl-CoA, and detoxification reactions. So these are the important roles of acetyl-CoA, and indirectly we can say pantothenic acid in the form of acetyl-CoA. So you can summarize the role of succinyl-CoA. It is coming from alpha-ketoglutarate, odd-chain fatty acid, methionine, and some of these amino acids, and utilized for TCA cycle, gluconeogenesis, porphyrins, activation of acetoacetate, and detoxification. So this way you can summarize the role of this coenzyme A in the metabolism. So sources of this pantothenic acid, as I have told, it is widely distributed in plants and animals, synthesized by normal bacterial flora in the intestine, and hence deficiency of this vitamin is rare. Yeast, liver, and eggs are the good sources of this vitamin. Recommended dietary alone. With ordinary diets, it can be made by 5 to 10 milligram. The deficiency manifestation of pantothenic acid, because of its wide occurrence, as such there is no noted pantothenic acid deficiency in the human who are on normal diet, but under strict experimental conditions it was studied, or it is found in animals where it can be developed only under experimental conditions in animal, that results in anemia, fatty liver, decreased steroid synthesis, etc. And in humans, it was Gopalan burning food syndrome, which is found to be due to deficiency of pantothenic acid, and this is manifested as paresthesia, that is burning, lightning pain in lower extremities and staggering gait. This was first time reported by the renowned nutritionist from India, that is Dr. Gopalan, who was later appointed as the Director General of ICMR also. He linked the burning feet syndrome, which was seen in the people who at the war and famine areas, in the soldiers of those who were deprived of the food. He found that the pain and numbness in toes, sleeplessness and fatigue in these people was because of pantothenic acid. And this syndrome, because of, in the honor of Dr. Gopalan, it is called as Gopalan's burning foot syndrome, which is due to deficiency of pantothenic acid. And it is seen during famine, in prison camps, and in chronic alcoholics, and in the patients who are on renal dialysis. So these are the points related to deficiency of pantothenic acid. You have to remember Gopalan foot syndrome. Assessment can be done by microbiological assay using Lactobacillus arabinosus, and this is found to be the structural analog of this, and that has got role as antivitamin, that is pantoyl taurine and omega methyl pantothenic acid. These are the two antivitamins of pantothenic acid reported. So with this, we come to the end of vitamin B5. Here you have to remember the role of vitamin B5 or pantothenic acid in the form of the activated derivatives of succinylate acid and acetic acid, that is succinyl-CoA and acetyl-CoA, and the deficiency manifestation as the Gopalan's burning foot syndrome. So with this quote, in the morning, one small positive thought in the morning can change your whole day. So with this, I come to the end of today's class. Next, Dr. Gohar ma'am will engage the class from 10 o'clock in my. Thank you.
Thank you, ma'am.
Yes. Now remaining amino acids we will come understand in later classes. Okay.